Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 2.5, membrane transport. Continuing with our series of subunits about the cell membrane, objectives today include describing the mechanisms that organisms use to maintain solute and water balance. So we're going to be talking about passive transport and active transport, as well as the more specific kinds of both of those general types of transport. And the suggested scale is going to be questions and methods, 3EB, propose a new or next investigation based on an evaluation of the design or method. So based on a current evaluation of passive or active transport, what could be a follow-up investigation for a certain scenario? So we're going to start by talking about passive transport. Cells do transport for the importing of materials and the exporting of waste, basically to keep the livable conditions inside the cell. And of course the cell has a membrane in order to create those livable conditions and separate them from the outside environment. So passive transport, also known as diffusion, is the movement of solids across the membrane from an area of high concentration to low concentration, so going with the concentration gradient. And it's basically how they would move naturally. So let's say we have a semi-permeable membrane here, meaning things can pass through it, at least to an extent, and we're gonna use small and nonpolar molecules because those can easily pass through the membrane. We're gonna put a lot of them on this side of the membrane. So as they're all bumping into each other, eventually they're going to spread out and disperse and move in this direction across the membrane until we've reached what's known as dynamic equilibrium. We have the same amount on each side, and they could be moving back and forth across the membrane, but there's going to be no net movement. They're not going to be moving all in one direction or another direction. It's not going to change the concentration. And here they're going with the concentration gradient because they're going from an area of high to low. This is passive transport because it doesn't require energy, because you're moving again with the concentration gradient. And there are two kinds. There's simple diffusion, which we showed here, small nonpolar molecules or small uncharged polar molecules, meaning water. And there's also facilitated diffusion, which is large polar molecules and ions, and these use membrane proteins. And all of 2.7 is about facilitated diffusion, so I want to talk about that much here. So now let's take a look at the diagram. So here's our passive transport diagram, and our goal is we're going to have movement across the membrane until we reach this dynamic equilibrium. Things can still be moving across the membrane. It's dynamic, but it's equilibrium, so overall we're going to have equal amount of solute on each side. So here's simple diffusion. We're literally just moving across the membrane from an area of high concentration, high amounts, to an area of low concentration, low amounts. And here are some examples of facilitated diffusion using membrane proteins. But the same thing is happening. These are all examples of passive transport. And what passive transport means, we're moving with the concentration gradient, high concentration to low concentration, and it's not taking up any energy. On the other hand, we have active transport, and this is the movement of solutes across the membrane against the concentration gradient, meaning we're going from a low concentration to a high concentration, and this is going to require ATP or some other form of metabolic energy, but usually ATP. And this can also use membrane proteins, which are going to be able to take resources in or expel resources when activated by ATP. You can also use energy from another solid's movement with the concentration gradient to power movement against. So let's say here's our membrane. We've got solids moving with the concentration gradient across the membrane, and the energy generated by this could power moving against the concentration gradient of this solid. And again, against the concentration gradient just means that we have a low amount here and we have a high amount here already, but we're still pumping things in, we're still cramming them all into the side where there's a high amount, even though naturally these molecules would be moving in the other direction. They'd be moving from high to low. And because we're going against the concentration gradient, against this natural flow, that's going to require the input of energy, or ATP. So let's look at a few specific kinds of active transport. So We looked on the previous slide, there was just active transport through a membrane protein. It was for few items, small solutes, and it's going to be a little bit of transport. Whereas here we have endocytosis and exocytosis, and these are going to be for large amounts of objects. And they're going to use the folds of the plasma membrane. And endocytosis, intake, endo, enter, and exo, expel. This is going to be getting things in and out of the cell using the folds of the plasma membrane, and it's going to take up large amounts. So there are a couple of types of endocytosis. First of all, we have phagocytosis, which is taking in large solid molecules by wrapping around the membrane, then pinching it off to turn into its own vesicle. So down here we have a picture of phagocytosis. The walls, or the folds of the plasma membrane, are coming up and around the solid particle. They're forming their own little vesicle, and all of a sudden that particle's been engulfed in the cell. In pinocytosis, they're taking in water and other substances, and this is through an inward fold in the membrane. We're able to suck in resources. So here's the membrane, we're going down, inward folding, and of course making our vesicle. So the difference between phagocytosis and pinocytosis are phagocytosis is going to be for larger solid molecules and the membrane folds are reaching up, while pinocytosis, membrane folds are going inward and we're generally dealing with fluid. Lastly, we have receptor-mediated endocytosis, which is going to be activating endocytosis only when the certain signal molecule binds to the cell receptor. It's going to activate the endocytosis when these star signal molecules are bound to the receptors on the cell, and that's going to form our vesicle. Now, exocytosis is, there are not many different kinds, but it's basically the same idea. We're just taking the vesicle that's already been created and we're opening it up through the membrane folds and letting all of the stuff out of the vesicle, out of the cell, and into the extracellular environment. So these are forms of active transport for large amounts of resources, endocytosis and exocytosis. Here we go, we're done with 2.6 membrane transport review. First of all, concentration gradients. We're going from high to low naturally, but with energy we can go low to high. You want to be able to know the different passive versus active transport, so simple diffusion, facilitated diffusion, endocytosis, exocytosis, and are they going with the concentration gradient or against? Are we using energy in the form of ATP or are we not? And lastly, we want to know the different kinds of endocytosis and exocytosis, such as phagocytosis, pinocytosis, and receptor-mediated endocytosis. Hope this was helpful. See you in facilitated diffusion.